Welcome to PW English YouTube channel. I'm Shalini Somashekar, your botany teacher. I welcome you to this lecture in the UMEED NEET English batch. Today we're going to start with the 12th grade syllabus, the first chapter, sexual reproduction in flowering plants. Before we start off with the concepts, I'll give you an overview of how many questions you can expect from this chapter in NEET 2025. As you can see, over the last few years, the numbers have varied between 1 and 5. Over the last few years especially, it has been roughly around 2 to 3. So that's around how many questions you can expect from this chapter in NEET 2025 as well. And talking about the syllabus, whether you should refer old NCERT or new NCERT, either of them will work. It is the exact same content in both. So whichever one you have, you can refer that for this chapter. So let's get started. As the name of the chapter itself indicates, here in this chapter, we're going to talk about how exactly sexual reproduction occurs in one group of plants called flowering plants. So, if you remember, in plant kingdom, we discussed different groups of plants. We spoke about algae, what are its different types, how does reproduction occur there. We spoke about bryophytes, pteridophytes, gymnosperms, and when it came to angiosperms, in the chapter Plant Kingdom, I told you we will not learn much about angiosperms in Plant Kingdom. There are 3 separate exclusive chapters meant for angiosperms. We've learnt about 2 of them already, morphology of flowering plants, anatomy of flowering plants. We've learnt about morphology and anatomy of angiosperms. In this chapter, we're going to focus on how sexual reproduction occurs in angiosperms. Okay, so angiosperms are nothing but flowering plants, right? So in this chapter, we're going to focus exclusively on how sexual reproduction takes place in flowering plants that are also called angiosperms. We know that all plants don't produce flowers. Flowers are the characteristic feature of angiosperms. You see it in angiosperm group of plants only. Now, flowers come in so many different colors, shapes, sizes. They're very pleasing to the eyes, and we have so many different ways in which we can make use of flowers. We use them to worship our God. We use them to express love. We use them in weddings. We use them in when somebody passes away, and if you want to mourn their loss, then you use flowers. We use them in home decor, and humans have come up with innumerable ways to make use of flowers. But why do you think plants produce flowers? Plants definitely, obviously do not produce flowers so that you and I can use them and, decorate our house or our hair or for whatever reason. The reason why plants produce flowers, especially angiosperms produce flowers, is because in angiosperms, flowers are the sexual reproductive organ. What do we mean when we say an organ is a sexual reproductive organ? For example, let's consider humans. We say that ovaries are the female sex organ and testes are the male sex organ. Why do we call them the sex organs? It is because in sexual reproduction, one very important process is the formation of gametes, right? Whichever part of the body formation of gametes takes place in, that part of the body is referred to as the primary sex organ, right? For us in females, ovaries produce the egg. So that is the female sex organ. In males, testes produce the sperms. So that is why it's called the male sex organ. Likewise, in plants, especially in angiosperms, for sexual reproduction, whatever gametes are required, they are produced in parts of the flower. Because they're produced by the flower, we call them the sexual reproductive organ. Okay. So in angiosperms, flower performs the process of sexual reproduction. That is why it's called the sexual reproductive organ in flowers. Now, how do flowers form on plants, right? So, if you think about how reproduction happens in humans, the moment a baby is born, it will not be capable of reproduction. It will undergo some period of growth, and then eventually, only later, around the time a child or a person hits probably 12, 13, 14, that year of age, that period of age, only then they will develop the ability to perform sexual reproduction. Likewise, in plants, the moment a seed germinates, it will not be capable of producing flowers. It will take a while for the plant to gain the ability to produce flowers. Initially, after the seed germinates, the plant will grow vegetatively for a period of time, and based on a lot of environmental cues and factors, the plant will decide when the time is right for flowering. Only when the conditions are favorable for flowering, flowers will be produced. See, by the process of flowering, seeds are going to be produced, right? Seeds are the next generation for the plant. So the flower should be produced at a time when if the seed is produced after the flower, then if the seed is lands on the soil and germinates, it should have suitable conditions for survival, right? Under such conditions only flowering should happen. Therefore, there are certain chemicals, certain pigments that are present in the leaf that can measure the day length, duration of day, duration of darkness during night, what is the temperature around. It can measure all of those environmental factors, and whenever it is suitable for flowering, only then it will initiate the process of flowering. It will send certain chemical signals from the leaf to the tip of the shoot. Once those chemical signals are sent, whatever was initially the shoot apical meristem it used to produce leaves, right now it will transform into a floral meristem. So at the floral meristem, instead of forming the shoot like the normal leaves and everything, what will actually form is a floral primordium, we call it, which will eventually become the flower. There an inflorescence axis will develop, and instead of leaves that are produced on the stem, floral buds will be produced. Eventually those floral buds will transform into flowers. Okay, so before you see the production of flowers on a plant, the decision that a flower should form would have been made, and a lot of processes and reactions and transport of chemicals will be taking place before you actually see the flower. All right, so much before the actual flower is seen on a plant, the decision that the plant is going to flower would have taken place, because the leaf measures everything, and once everything is favorable, it would have decided that it is time to reproduce and produce flowers. At that time, several hormonal and structural changes will be initiated. I told you, right? Chemicals will be transported from the leaf to the apical meristem. So all of these things will keep happening, and it leads to the differentiation and development of the floral primordium. Okay. So from here, inflorescence will be formed. In the inflorescence will bear floral buds, which will eventually become flowers. We know from the chapter Morphology of Flowering Plants that flowers are modified condensed shoot. Flower is a modification of the stem. Whenever stem is performing anything other than its usual primary functions, we say that it is modified. Over here, flower is a modified shoot because it's modified for the purpose of sexual reproduction. We are saying it's a condensed shoot because in a flower, the internodal length is highly reduced, and all the whorls will appear to arise from the same point. So the shoot is condensed. About that I've already told you in the chapter Morphology of Flowering Plants. So I hope if you've studied that you would remember. Alright, so that's the thing about initiation of flowering. Now let's talk about the different parts of a flower. We've already discussed flower in detail in the chapter Morphology of Flowering Plants. What we already know is that the stalk of a flower, if it is present, is known as the pedicel. At the tip of the pedicel, you have a broad region, flat and broad region, which is called the thalamus. On the thalamus, different parts of the flower are arranged in form of concentric circles, which we call the whorls. The outermost whorl is called the calyx, and it is composed of small green leaf-like structures called the sepals. Inside that, you have the corolla, which consists of the petals, which are usually the brightest colored, most attractive part of the flower. And inside that, you have the gynoecium, which is the female, Inside that, you have the androecium, which is composed of the stamens, which is the male reproductive part of the flower. And right within, in the innermost whorl, we have gynoecium, which is the female reproductive part. It has units called carpels that it is composed of. Since over here we are talking about sexual reproduction specifically, our focus will mostly be on these two whorls. This is the female reproductive part, this is the male reproductive part. It is within the stamens that the male spores will form. It is within the carpels or the gynoecium that the female spores will form. So we will focus mostly on these parts of the flower. About the other parts, its variations and everything we've spoken in the chapter Morphology of Flowering Plants. Alright, so let's get started with how sexual reproduction takes place in flowering plants. You know that in sexual reproduction, one of the most important features is the fusion of gametes. The process by which fusion of gametes takes place is called fertilization. In this chapter, we're going to learn about how sexual reproduction happens in three different parts. In the first part, we learn about what changes take place before fertilization occurs. We call them the pre-fertilization structures and events before the process of fertilization. For fertilization to happen, gametes have to form, right? So how do the gametes form? How does the gametophyte develop? All of that we learn in the pre-fertilization structures and events. After that, we learn about how exactly fertilization takes place in angiosperms. There is a characteristic type of fertilization called double fertilization that occurs. How exactly it takes place, what happens there, how is it different from other types of fertilization, we'll learn about it in that. And after fertilization, whatever changes take place, whatever events take place, whatever structures form, about that we learn under post-fertilization structures and events. So the three parts are pre-fertilization events, fertilization, how does it take place, and post-fertilization events and structures. Alright, before we start off with how pre-fertilization structures form, I want to take you back to the life cycle that we see in plants, the life cycle of plants, which exhibits alternation of generation. We've learnt about it in the chapter Plant Kingdom, but I want to remind you of it again. So in angiosperms, the main plant body that you see is sporophytic, which means it is diploid. It has two sets of chromosomes, right? It is diploid in nature. A sporophyte forms spores. So within the sporophyte, the male spore, which is the microspore, and the female spore, the megaspore, will form by a process of meiotic division. The spores are haploid. So in the sporophyte, due to meiotic division, male microspore and female megaspore will form. Spores will develop into the gametophytes, right? So the spore will undergo mitosis to form the male gametophyte. Male spore forms the male gametophyte. Female megaspore forms the female gametophyte, which is haploid. It is the job of the gametophyte to give rise to the gametes. So within the gametophyte, again, mitosis happens and the male and female gametes are formed. Once the male and female gametes fuse by a process of fertilization or syngamy, whatever you call it, the zygote will form. So diploid condition is restored after fertilization. This zygote will develop into the diploid sporophyte. So I want you to notice how the sporophytic phase is alternating with the gametophytic phase. Alright. So over here, the formation of microspore happens in the stamen, more specifically in the anther, and the formation of the megaspore happens within the ovule. Okay, how exactly it happens? We'll learn about that. First, starting with how the microspore forms. So I mentioned already in this chapter we're focusing on how sexual reproduction will take place. So our focus will be mostly on the reproductive parts, andrecium and gynoecium. Andrecium is the male reproductive whorl. It is composed of units called stamens, and stamens typically have two parts. It is composed of a long slender structure called the filament. The filament on one end, on the proximal end, is attached to the thalamus. On the other end, it bears a lobe-like structure, right? A knob-shaped structure, which is called the anther. The anther generally in most angiosperms consists of two lobes. Okay, so if you see here, if you cut it like this in this plane and look at it, it will appear like this. Over here, this is one lobe, and right here you have the second lobe. These two lobes are connected by what we call the connective. Since the anther contains two lobes, we can say that the anther is bilobed. If you now look at each lobe here, if you can think of this as one lobe, look at each lobe here. Within each lobe, you can find two cavities or chambers that run throughout their length longitudinally. So this is one and this is the other. Within each lobe, you can find two of them. These cavities or chambers are called theca. So in each lobe, you have two theca. So we can say it is diathecous. It is right within these chambers that we have here. Inside this, microspores will form. The structure within which spores form is called the sporangium. Over here, within these structures, microspores will form. So these structures can be called as microsporangia. There's a total of 4. 1, 2, 3, 4. 4 microsporangia present in each anther. Each anther has 4 microsporangia. So we call it tetrasporangiate. Tetra here means 4. Di means 2. Bi also means 2. Because there are 2 lobes, it's bilobed. Because each lobe has 2 theca, it is diathecous. Because each anther typically, in most cases, has 4 microsporangia, it is tetrasporangiate. In between 2 theca, in the same lobe, there will be a groove-like thing that is called the stomium or the line of dehiscence. After the pollen grains are formed inside the anther has to break open to release the pollen grains, right? So the pollen grains are released when the anther opens. The anther opens specifically in this groove. So it opens, splits open over there. That is why it's called the line of dehiscence. Dehiscence basically means opening of anther to release pollen. Okay, now let's talk about the structure of one microsporangium here. There is 4 of them in the anther. As you can see here, this is 1, this is 2, this is 3, this is 4. It's right within this microsporangium where pollen grains are produced. So microspores or pollen grains are very, very important because they will give rise to the male gametophyte, and that is what will form the male gametes for the continuation of generation. This is very important, very, very precious, important kinds of cells are going to be produced here. It requires good amount of protection. So the wall of the anther that is protecting this tissue, which is in future going to give rise to pollen grains, is very, very protective. It has many different types of layers present in it, each layer having its own benefits to offer. Like similar to how we have walls in our homes, walls in general are supposed to offer protection. Here also the wall of the anther does offer protection, but each layer has its own function to perform. Similar to how in our walls there is a brick layer in the center which will provide that mechanical strength. On top of that you have cement which will keep it together, and then on top of that you probably have primer, on top of that you have paint or wallpaper. So each layer in your wall serves its own purpose. Like that, the different layers in the wall of the anther have their own purpose to serve. Here you can see the wall is pretty thick. So let's learn about what are the different layers that a wall of the anther is composed of. The outermost layer is epidermis. It is usually 1 cell layer in thickness. You know how in every part of the plant, be it root, stem or leaf, the outermost layer is always the epidermis. Even over here it is the epidermis. It is composed of barrel-shaped cells with no intercellular spaces. It's kind of like a compact layer. It offers protection. Secondly, we have something called endothecium. Endothecium also is just 1 cell layer in thickness. Okay, and here the endothecium, this also offers protection when the anther is young. Inside that we have the middle layers, which is 1 to 3 cell layers in thickness. Okay, now all of these 3 layers, whenever the anther is young, it will offer protection. that is when it needs protection, right? Because inside the anther, very special cells, very precious cells are being formed. So that needs protection. But once the anther matures, pollen grains would have already been produced. After pollen grains are formed, we say that the anther is matured. Once the anther matures, it is important for the anther wall to break apart. It shouldn't, see, the wall layer shouldn't be always intact and protecting the pollen grains. Of course, whenever protection is required, it has to offer protection. But when it is time for the pollen grains to be released, then the anther wall has to break open and release the pollen grains. If it is like a protective wall throughout, then even after the pollen grains have formed and the anther is matured, if the wall is intact and protecting still, then the pollen grains cannot be released. If the pollen grains are not released, how does sexual reproduction in plants even take place, right? So whenever the anther matures, the same three layers will help in dehiscence of anther, it will help in breakdown of the anther wall. It helps in dehiscence. Now the innermost layer in the wall, okay, from here to here is the wall. This is the epidermis, this is the endothecium. Here you had two layers of middle layers and here you have the tapetum. Tapetum is not protective in function. It basically, okay, this also generally is single layered. Here you can find multinucleate cells. Okay, so this basically nourishes the developing microspores. Okay. See, I want you to understand that whichever region I am outlining right now, whatever cells are present over here, these are called microspore mother cells, abbreviated as MMC or even sometimes as pollen mother cells, abbreviated as PMC. All right. Now, what does microspore mother cell mean or pollen mother cell mean? It is these cells which are eventually going to give rise to pollen grains or microspores. So it is these cells which are directly involved in the formation of pollen grains. During the formation of pollen grains, nourishment is required. So the cells that are present immediately surrounding it are going to provide nourishment to these cells. Okay, so the tapetum is the innermost layer of the anther, innermost layer of the wall of the anther, which is closest to the microspore mother cells. So this will provide nourishment to the developing microspores. Alright, so these microspore mother cells or pollen mother cells, these are eventually going to form microspores or pollen grains. Alright, now let's talk about how exactly microspores form. The process by which microspores form in the anther is called microsporogenesis. Over here, microsporo relates to microspore and genesis means generation, or you can call it formation, right? So, microsporogenesis can be thought of as the process by which microspore mother cells undergo meiosis to form haploid microspores. It's a very simple process. Let's just consider the sporogenous tissue or microspore mother cells in the center. Okay, so within the anther there will be four microsporangia. Inside each microsporangium you can find many such microspore mother cells, similar to how all neighboring plant cells will have plasmodesmatal connections between them. Between these microspores also you can find, between these microspore mother cells also you can find plasmodesmatal connections, cytoplasmic connections. The first thing that happens during microsporogenesis is a callose wall will start forming around each cell such that all of those cytoplasmic connections are cut off. All right. Now we have a lot of microspore mother cells with no connection to its neighboring cells. Okay. Let's just consider one microspore mother cell that is diploid. This will undergo meiosis. As a result, as a product of meiosis, you will get 4 cells and ploidy will be reduced to half. So from one microspore mother cell, we will get 4 microspores. You can see 1, 2, 3 here, the other one's behind. All right. So there are 4 microspores formed. Since there are 4 microspores formed, we call this a microspore tetrad. Eventually, when they're formed, all of them will be present within the same callose wall itself. Eventually, because of some secretions of the tapetum, the callose wall that was present around will dissolve and each of these cells will start separating out and the 4 cells will separate out, at which point we call them the pollen grains. The pollen grains are haploid in nature. All right. Now, that's about how the microspores or pollen grains form. Generally, students think that pollen grains themselves are the male gametes, but the pollen grains are not the male gametes, they are the male gametophytes. Remember, we've learned in the life cycle of plants, alternation of generation, that spores will give rise to the gametophyte. Gametophyte will produce gametes. Right now, this spore, microspore, has given rise to the gametophyte, the pollen grain. Within the pollen grain, male gametes will be formed. So, gametes are formed inside the pollen grain. The pollen grain itself is not the gamete. Generally, pollen grains are spherical in shape. All right. They come in many different shapes, but majorly, they are spherical in shape. And their size is very, very small. They measure around 25 to 50 microns in diameter. Okay. Talking about its structure, a pollen grain is composed of 2 wall layers. The wall of the pollen grain has 2 layers. Okay. An outer layer which is called the exine, and there is an inner layer which is called the intine. Okay. There is an outer exine and inner intine. What I've represented in orange is the exine. All right. Orange is exine. Exo means outside. So the outer layer of the wall of pollen grain is called the exine. Exine is made up of a substance called sporopollenin. This is one of the toughest biological materials known. It is resistant to strong acids, alkali, high temperature, and even enzymes. We know no enzymes that can degrade sporopollenin. It is composed of a very tough material that is very essential because inside it's carrying male gametes that requires protection. So the sporopollenin is very, very hardy and a resistant material. Now this exine, as you can see, is a discontinuous layer. Sporopollenin is not uniformly present throughout the wall. There are some regions where sporopollenin is absent. They are called germ pores. In these regions, sporopollenin is absent. Alright. Now, what do we have inside? The inner wall layer is called intine. This intine is just like a regular plant cell wall. It is made up of cellulose and pectin. It is relatively thinner compared to exine and it is a continuous layer. You do not have any pores in the intine. Initially, when the pollen grain is formed, you have only one haploid nucleus, one haploid cell present inside it. But soon that single cell inside the pollen grain there will divide, undergo mitotic division to give rise to two cells. That mitotic division will be unequal, it will not be uniform. So it will result in the formation of a large cell and a small cell. The large cell is called the vegetative cell. It will have abundant reserve food present in the cytoplasm. It will have a very large irregular nucleus as well. In the vegetative cell cytoplasm, the relatively smaller generative cell will keep floating. Okay, so this is the generative cell. It has a small nucleus and this whole cell is spindle shaped. This floats in the cytoplasm of the vegetative cell. When the pollen grain has two cells, one the vegetative cell and the generative cell, these two cells present, the pollen grain is said to be in the two-celled stage. Alright. Now, I've already explained all of this. The same thing is written in text in your notes for your reference. I've already told you about the vegetative cell and generative cell as well. So, that also is mentioned here. Vegetative cell is larger, has abundant food reserve, large irregularly shaped nucleus. Generative cell is small, it floats in the cytoplasm of the vegetative cell. It is spindle shaped, it has dense cytoplasm and nucleus. That is about the two-cell stage. But the two-cell stage of pollen grain is not its mature stage. The pollen grain will have three cells in its mature phase or mature stage. I told you that the pollen grain is the male gametophyte. That means it should produce male gametes. But right now, so far, I haven't labeled any part as, the male gametes. That means the male gametes haven't yet formed, right? There are germ pores and everything. What basically happens is, this large generative cell that was there, it will not undergo any change. The large vegetative cell. The generative cell will undergo mitosis and give rise to two male gametes. Okay. So over here in the three-celled stage, you will for sure have the large vegetative cell with its abundant food reserve and cytoplasm and large nucleus and everything. But the generative cell that was small and there will no longer be there. It would have undergone mitotic division to give rise to two small male gametes that also float in the cytoplasm of the vegetative cell. Whenever within the pollen grain there is one vegetative cell and two male gametes and there's a total of three cells, you call it the three-celled stage. In the three-celled stage, you have one large vegetative cell within which there are two Male gametes that are small and floating. Overall, in the pollen grain, you have 3 cells right now. And this is the mature pollen grain or gametophyte, because right now it has fulfilled its job to produce the male gametes. Alright. Now, this is the diagram that is given in NCERT. This is the pollen grain that was initially formed. It is very small in size. It will undergo vacuolation. Vacuoles will form. There will be unequal cell division that takes place. See, spindle formation is not symmetric, it is asymmetric. As a result, a large vegetative cell and a smaller generative cell will form. Alright. So, up until this point, we've learnt about how pollen grains form, and from the pollen grains, within the pollen grains, how the male gametes will form. Pollen grains have certain advantages and disadvantages as well. First, let's talk about what are some disadvantages of pollen grains. Of course, they are very beneficial for plants because they bring about sexual reproduction. But pollen grains of some plants can cause severe allergies in humans and other animals. It can cause a lot of respiratory tract infections. It can cause asthma and bronchitis and all of those things. Because they're very small dust-like particles, when they're inhaled, it can kind of inflame your respiratory tract, right? So pollen grains of several species cause allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders like asthma and bronchitis. One of the plants which is known majorly to cause pollen allergy is Parthenium. It is called Congress Gida in Kannada. I don't know what you call it in Hindi and other languages. Okay, so it is called Parthenium. It is known to cause pollen allergy. Now, talking about the advantages of pollen grains, they are made up of a lot of nutrients. So they are rich in nutrients. Humans can, it's available for humans in the form of tablets and syrups. So that can be consumed to, enhance your nutrition. All right. And it is also believed that in athletes and race horses, pollen tablets are provided in order to make their performance better. So these are some advantages of pollen grains. Now, what is the job of pollen grain? Pollen grain's job is to carry the male gamete to the female reproductive part and make sure that the gametes reach where they need to reach and bring about fertilization. For that, pollen grains have to fall on the stigma and form a pollen tube. Basically, they have to germinate on the stigma and form a pollen tube. For as long as pollen grains retain the ability to form a pollen tube to germinate on reaching compatible stigma, we say that the pollen is viable. As long as it's capable of doing its job, we say that the pollen is viable. Pollen grains do not retain viability forever. So, across species, across different families, for how long a pollen grain can be viable varies, okay? In some species or in some families like wheat, and other grasses, you can see that the pollen loses its viability at around 30 minutes. But in some families like Solanaceae and Rosaceae, you can see that for a few months, the pollen viability will be retained. Okay. So once the pollen grains are released or shed, they have to land on the stigma before they lose their viability to bring about fertilization. The viable period is variable. It depends on a lot of factors like temperature and humidity, of course. And in some cereals like rice and wheat, within 30 minutes of release, they will lose their viability. And in members of Rosaceae, Leguminosae and Solanaceae, it can last for a few months. For crop breeding programs, it is possible to store pollen grains and preserve them in liquid nitrogen at minus 196 degrees Celsius, which we refer to as cryopreservation. When pollen grains are preserved like that, we call them pollen banks. Okay, so they can be stored in liquid nitrogen, specifically at minus 196 degrees Celsius. So their viability can be, preserved for a longer period. All right. Now, let's talk about the female reproductive part. So far, we've discussed what happens in the anther, how the pollen grains form, how do the male gametes form within the pollen grains. So the story about males is done. We have the male gametes ready. Now we have to get the female gametes ready so that fertilization can take place. The female reproductive part is called the gynoecium or the pistil, and the units of gynoecium are called carpels. In some plants, there is only one carpel. In some plants, there are many carpels. When there is only one carpel present, we call that monocarpellary condition. Okay, so here only one carpel will be present. Whenever there is more than one carpel present, we call it multicarpellary condition. Whenever there's more than one carpel, the carpels may be fused or they may be free. If they are fused, we call it syncarpous. If they are free, we call it apocarpous. Example for syncarpous condition is papaver. Example for apocarpous condition is Michaelia. Michaelia is sampige, champaka I think it's called, I'm not sure in Hindi. Sampige, yellow color, yellowish orange color flowers it produces. So there it is apocarpous, not fused, and in, apocarpous is here, this is Michaelia. And the syncarpous is papaver. You can see that the different carpels are fused here. Alright, examples, very important, you must remember. Now, in a gynoecium, you find three parts. There is the stigma, the style, and the ovary. Stigma is this top flat broad region that you find, which is where the pollen grains will come and land. So it is the part of the gynoecium which receives the pollen grains. So this acts as a landing platform for the pollen. It is kind of sticky so that the pollen grains that land on it stay right there. Second, we have the style, which is kind of boring, doesn't do much. It is something that is slender and thin and long and connects the stigma to the next part, which is the ovary. Ovary is broad, bulged, swollen. Within the ovary, you will have a structure, cushiony structure called the placenta. And to the placenta, many ovules, one to many ovules will be attached. Okay, so this is the basal part of the gynoecium. So this is what the ovary looks like. As you can see, there is a wall, the ovary wall. Inside that, there is a cavity which we call the locule. And inside the locule, here you have this fat placenta. To the placenta, ovules are attached. Ovules are nothing but the megasporangia. It is within the ovules that megaspores get produced. Okay, depending upon the species, within the ovary, one ovule may be present or many ovules may be present. In fruits like mango, there's only one ovule per ovary. In fruits like tomatoes, many ovules are present. Watermelon, many ovules are present, right? And also in monocots like wheat and paddy, there's only one ovule per ovary. Now, let's understand the structure of ovule or the megasporangium. It has a stalk that we call the funicle. I will outline it for you over here. So this is the stalk of the ovule. Okay, so this is the stalk of the ovule. The stalk is called the funicle. This whole thing that I've outlined here is the body of the ovule. The stalk attaches to the body. It's, joined or fused with the body at one point. That point at which the stalk fuses with the body is called the hilum. Similar to how microsporangium was important because the male spores used to get produced inside and it required protection. Here, inside this megasporangium, megaspores or the female spores are going to get produced, so that also requires protection. Protection is provided by two integuments. Here you have the outer integument that I've just outlined, and here inside that you have the inner integument. Okay, so you have outer and inner integuments that are protective in function. Inside the integument, you have a homogeneous mass of cells, parenchymatous, that is called the nucellus. It is within the nucellus that the female megaspore will get produced, and the megaspore will develop into the female gametophyte inside the nucellus itself. The female gametophyte is also known as the embryo sac. As you can see, the two integuments do not fully cover the nucellus. There is a small region where there is an opening. That opening is called the micropyle. The part of the ovule or the embryo sac towards the micropyle is called the micropylar pole. The opposite pole is called the chalazal pole or the chalazal end. These are the different parts of a megasporangium. Now we have to learn about how exactly megasporogenesis or development of megaspore takes place inside this ovule. Alright, so here you know how in flowering plants the ovule itself will develop into the seed. In the seed you can see there is a small opening called the micropyle. This is the hilum. I'm sorry. Yeah, that part where it fuses with the body, that is the hilum. Okay, here you can see this is the opening micropyle. The two integuments will eventually in the seed become the seed coats, outer integument testa, inner integument tegmen. More about it we'll learn later. Now let's understand how megasporogenesis takes place. I told you how inside the integuments there is homogeneous mass of cells called the nucellus. Initially, when the ovule is not mature, very young, all of the cells will look alike. But as the ovule starts to mature, one of the cells of the nucellus will start appearing different from the other cells. It will get differentiated. It will become larger in size compared to the other cells. I want you to notice this cell and compare it with its neighboring cells. Look at how it is also large. The nucleus is also larger. It has a dense cytoplasm and prominent nucleus. That one nucellar cell that differentiates from the other surrounding nucellar cells is called the megaspore mother cell. Generally, it is produced towards the micropylar end of the ovule or of the nucellus. In microsporogenesis, we saw how every one of the cells in the sporogenous tissue had the ability to form microspores. In megasporogenesis, only one nucellar cell that differentiates will form the megaspore mother cell and give rise to the megaspores. So, this megaspore mother cell will undergo first meiotic division to give 2 cells, and then after the second meiotic division, obviously we will get 4 haploid cells. They are the megaspores. These megaspores, there's 4 of them, so we call it the megaspore tetrad because there's 4 of them and they are haploid, right? Of these 4 megaspores, 3 megaspores will degenerate and only 1 will remain functional. This one functional megaspore will develop into the female gametophyte, which we also call the embryo sac. Since a single megaspore is involved in the development of the female gametophyte, we call it monosporic development. Okay, we call it monosporic development. What is megasporogenesis? Megasporo relates to megaspore. Genesis relates to generation or formation. Megasporogenesis is the process by which megaspores form. It happens within the ovule. It involves meiotic cell division. Alright. Now, megaspore is formed. From the megaspore, how does the female gametophyte develop? We will understand that. We have just one megaspore which is haploid. The single megaspore undergoes mitosis. After the first mitotic division, 2 nuclei are formed. After this mitosis, karyokinesis happens during mitosis. Karyokinesis is immediately not followed by cytokinesis. So within the same cell, you will have 2 nuclei. These 2 nuclei will migrate to the opposite poles. And once they've moved to the opposite poles, another round of mitosis will take place. This is the second round. After which, at each pole, you have 2 cells. After this, the third round of mitosis will take place for the third time. And you will have 1, 2, 3, 4, 1, 2, 3, 4. 4 nuclei at each pole. Again, in all these mitotic divisions, karyokinesis is immediately not followed by cytokinesis. Cell wall formation does not happen. So all of these nuclei are present within the same cytoplasm. Once the third mitotic division takes place, one nucleus from each pole migrates to the center and cell wall formation begins. As a result, the mature embryo sac will form. In the mature embryo sac, you have two cells in the center that have moved from the opposite poles. At each pole, you will have three cells present. Okay. Because cell wall formation has happened right now, I'm calling it cells and not nuclei anymore. There are three cells at each pole and two nuclei at the center. The cells that are present at the chelazal end, these three cells are known as the antipodals. The cells that are present towards the micropylar end, these three cells together constitute the egg apparatus. The central cell here in the egg apparatus is the egg cell, which is the female gametophyte. On either side of the female gametophyte, you have cells that are called synergids. Synergids have cellulosic thickenings towards the micropylar end that are called filiform apparatus. This whole large cell in the center is called the central cell. Within the central cell, you have two haploid nuclei that are called the polar nuclei because they migrated from opposite poles. Alright, so this is the final structure of the female gametophyte or the embryo sac. This is the diagram that is given in NCERT. Chelazal end you have antipodals, micropylar end you have the egg apparatus, central cell, polar nuclei, all of that. Alright, now pollen grains are produced inside the female reproductive structure, inside the megasporangium specifically. An embryo sac is formed, right? Remember that the pollen grains will be released. Male gametophyte is released, but the female gametophyte or the female spore will not get released. It is retained within the sporophytic plant body itself. It is present within the ovary itself. Now the pollen grain has to be transported, and it has to go and land on the stigma and bring about fertilization and all of that. For that, pollen grains have to be transferred from the anther to the stigma. The process by which the transfer of pollen grains happens from the anther to stigma is called pollination. It can happen in many types. Alright, there are three distinct types of pollination based on the source of pollen. There is autogamy, geitonogamy and xenogamy. We learn about each of them one by one, starting with autogamy. Autogamy is nothing but self-pollination. The anther of one flower produces pollen grains. If the pollen grains that are produced by the anther of one flower land on the stigma of the same flower, we call it autogamy. Over here, one plant and one flower only is involved. For autogamy to take place, there should be synchrony between when pollen grains is released and when stigma becomes mature. When the pollen is released at the same time if stigma is mature, then autogamy is possible, and for autogamy to take place, the flower has to be bisexual. If it is unisexual, it's not possible, obviously, right? So it requires the flower to be bisexual, and synchrony has to be there. It has to happen at the same time. What has to happen at the same time? The release of the pollen grains and the stigma becoming mature and receptive. Also, the anther and stigma should lie very close to each other so that pollination can be brought about. Okay, now there are some species of plants like viola, camelina and oxalis in which two types of flowers can be found within the same plant. One type of flower blooms, exposes the anther and stigma, that is called chasmogamous flowers. There is another type of flowers which does not bloom, which is always closed. It does not open and expose the anther and stigma. Such closed flowers are called cleistogamous flowers. Here you have chasmogamous flowers, here you have cleistogamous flowers. So here you can see, this is the camelina plant. Here you have chasmogamous flowers, here you have cleistogamous flowers. This is not cleisto, This is 𝐄𝐍𝐆𝐋𝐈𒀻. Okay. So what happens in 𝐄𝐍𝐵𝐋 flowers is the flower is open. So chances are there for self-pollination, but it's not 100% sure that self-pollination only will happen because pollen grains are, the anther and stigma are exposed. It's possible for pollen from these flowers to go and fertilize or bring about pollination in some other flower also, right? In contrast, if you consider the 𝐒𝐍𝐵𝐋 flowers which never open, over here, since they don't open, there is no chance of this pollen in this flower leaving the flower and going and pollinating some other plant or some other flower. Also, since this flower doesn't bloom at all, there is no chance of some other pollen coming and landing on the stigma of this flower. Since they are always closed, doesn't open, the anther and stigma are not exposed. Inside the floral bud, you have stamen and the stigma very close to each other. It mostly undergoes self-pollination, not mostly, it always undergoes self-pollination or autogamy, okay? And that is why we say they are invariably autogamous, 100% of the time autogamous. There is an advantage to being 𝐒𝐍𝐵𝐋. There 100% of the time you can ensure for pollination to take place. After pollination, of course, fertilization will take place. If fertilization takes place, then seed formation will take place. They don't have to depend on any external agents like insects or wind to bring about pollination, since they are very close to each other and they undergo autogamy. Seed set is assured 100% of the time. These flowers will produce seeds. Okay. Now the second type is called 𝐵𝐻𝐻𝑔amy. Over here, one plant is involved, two different flowers of the same plant is involved. Over here, the flower may be unisexual or bisexual. So whatever stamens are there in one flower, they release pollen grains. The pollen grains that are released, if it goes and lands on the stigma of another flower belonging to the same plant, then we say it is 𝐵𝐻𝐻𝑔amy. Genetically, if you think about it, this is also like self-pollination because it's coming from the pollen grains belonging to the same plant. But technically, if you see, it is cross because it's happening between two different flowers, right? So functionally, it is cross-pollination. Genetically, it is self-pollination. The last type is called xenogamy. This is actual cross-pollination where two different plants are involved. So genetically, different types of pollen grains can be exchanged. Whatever pollen grain this plant produces, that if it goes and lands on the stigma of another flower stigma of another plant, then we call it xenogamy or cross-pollination. So this is the only type of pollination where genetically different types of pollen grains are brought to the stigma. These are about different types of pollination based on the source of pollen. Okay. Now we have to understand how, if cross-pollination happens, by what agency is the pollen grain getting transferred? Because pollen grains don't have locomotory structures, they don't have legs to walk, they don't have cilia flagella. If they have to be carried to a different plant or a different flower, then it has to happen by some external agency, either wind, water, bird, insect, whatever. If the external agent is non-living like wind and water, we call it abiotic agents. If it is living like insect and bird, then we call it biotic agents. Abiotic agents, wind and water, this is rarely found in plants. More common one is animals. Among animals, insects. Among insects, it's bees, that is most common. Okay, we learn about each of these types starting with so let's talk about this. The number of pollen grains that are produced in plants is much higher compared to how many embryo sac is present or how many ovules are present in the ovary. Similar to how the number of sperms is produced that are produced in humans is much higher compared to the number of eggs that females produce, because the chances of fertilization will be very less if fewer sperms are produced, because sperms have to travel a lot of distance and everything. Likewise, even for pollen grains, just because a pollen grain is produced and released doesn't mean it will find a compatible stigma and bring about pollination, fertilization and all of that. So to increase the chances of successful pollination and fertilization, the number of pollen grains that are produced is very, very high. Ultimately, it is a chance factor only. Okay. Let's talk about wind pollination, which is also called anemophily. We have to learn about how in plants that undergo wind pollination, pollination by wind, how are the flowers modified there? If it undergoes water pollination, how are the flowers or the plant modified there? If it undergoes pollination by insects or living organisms, birds, whatever, how is the flower modified in that way? Okay, so if you're talking about wind pollination, wind is an abiotic factor. It cannot see, it cannot smell, it cannot taste. So it's not required for the plant to produce energy to look good, because wind cannot see. Even if the flowers are brightly colored, wind cannot see, it doesn't care. Even if it's producing nectar, if it's producing nectar at all, then the wind cannot eat it, right? If it is giving out like a fragrance, then the wind cannot sense it, right? So this plant should not waste energy in producing brightly colored petals, producing fragrance, producing nectar and all of that, because for wind it doesn't matter, right? So over here, what plants do is, in order to make sure that whatever pollen grains are released in the wind are carried by the wind to long distances and bring about effective cross-pollination, the pollen grains that are produced will be very, very light. They'll not be heavy at all. They will be very light and they will be non-sticky also, so that the wind can be transported off to long distances. That is about the pollen grains. How are the anthers then? If the anthers are present, right inside, hiding inside the petals, then after dehiscence of anther, pollen grains cannot, it is difficult for pollen grains to be carried by the wind because it will settle right inside the flower. Therefore, in such plants, the anthers are exposed, they are outside, and whenever pollen is released, it's easier for wind to carry it forward. So that is why the anthers are exposed. Pollen grains are light and non-sticky. Talking about the stigma is very feathery in nature, so that it can easily trap the airborne pollen grains. All right. And flowers are also not usually solitary, they're not single. Many flowers are packed in an inflorescence. Wind pollination we generally see in the family of grasses like wheat, maize, rice, ragi and all of that. So you can see how the inflorescence is in these plants. It is not very colorful and bright, right? So they are very pale kind of colors. But they are packed together in an inflorescence. Pollen grains are light and non-sticky. Large amount of pollen grains is produced in all types of plants, even over here. Stamens are well exposed so that they can be dispersed easily. Stigma is large and sticky and feathery. All right. Generally in these plants, per ovule there will only, per ovary there will only be one ovule, and like I already mentioned, this is seen in grass family, Gramineae or Poaceae. You've seen, if you've all eaten corn, I'm sure you know that these silky hair-like thing that you see, the corn tassels in the cob, they are nothing but the exposed style and stigma. All right, now let's talk about water pollination. Just because a plant is aquatic doesn't mean it will undergo water pollination. Even if the plant is aquatic and the flowers are produced at the surface like lotus and water lily, pollination can take place by wind or insect as long as the flower is exposed on the surface of water. Only if the flowers are produced inside the water, pollination happens by water. This is the rarest form of pollination. In angiosperms, only around 30 genera you can see water pollination. Among them also mostly it is monocotyledonous plants only. So yeah, examples we have in freshwater forms Vallisneria and Hydrilla, and in marine forms we have Zostera or sea grasses. Alright, how exactly pollination occurs here? Let's understand. First, let's see how it happens in Vallisneria. Vallisneria is a dioecious plant. You have a separate male and a female plant. Male plant produces the male flowers or the pollen grains. Female plant produces the female flowers. The male flowers that are produced by the male plant will get released outside and they will start floating on the surface of water passively. The female flower will not get released. The female flower will have a very long coiled stalk like this. Okay, so this is the flower. This is its stalk. Okay, initially it will be coiled and present inside the surface of water. Once it matures, it will rise to the surface of water. In the surface of water, male flowers will start floating. See, this is the female flower. These small things are the male flowers. You can see that it is floating passively. If by chance male flower reaches the stigma, then pollination will happen, fertilization also will be brought about. After that, this flower will coil back down and come to the water. Inside the water, it will just go back to water, and within the water itself, fruits will be produced. So that is how pollination happens in Vallisneria. If you have to talk about sea grasses, over here, this is a monoecious plant, but the same plant will produce separate male and female flowers. Okay, so it is unisexual flowers but a monoecious plant. Okay, so here you can see how the male gametes that are produced by the male flowers, the pollen grains, I'm sorry, not the male gamete, the male gametophyte, it will have long ribbon like shape. Over here, the pollen grains are not released to the surface of water. They will be present within the water itself. Even the female flower will not reach the surface, it will be present within the water itself. The pollen grains, because of having a long ribbon-like shape, it can float passively within the water itself and reach the female flower and bring about pollination and eventually fertilization. Okay, so over here female flowers will remain submerged. Pollen grains are also released inside the water only, they are not going to the surface. So pollen grains are long and ribbon-like, which will help it passively float inside water itself. If it reaches the stigma, then it will achieve pollination. Okay, in most of these plants which will undergo pollination by water, there is a mucilaginous covering that it will have, the pollen grains, that will prevent it from getting wet. Okay. Now let's talk about animal pollination, which is also called zoophily. Generally, when we talk about pollinators, animal pollinators, we think about insects like bees and butterflies. But there are many groups of animals that bring about pollination in nature. Of course, what we know about commonly are bees and butterflies, flies also, beetles, wasps, ants, moths. These are common insects. But along with them, birds bring about pollination, bats, primates like lemurs, arboreal, tree-dwelling animals, rodents like mice or reptiles like lizard also can bring about pollination. Insects, mostly bees, are the most dominant biotic pollinating agents. In majority of plants, bees only are the pollinating agents. If a plant has to be pollinated by insect, they have to attract the insect somehow. Insect is an animal that has senses. It can see, it can smell, it can taste. So whatever is required to attract an insect, a flower has to do that. And also, in order to make sure that insects keep coming back to your plant to bring about pollination, we should also offer some reward. Every time you come, I'm going to give you something so that it makes you come back to the flower. Now, what are the attractants? How can a flower attract an insect? By being colorful. Like previous two types, abiotic pollinating agents, they cannot see, they cannot smell. So if a plant spends energy in being beautiful, producing pretty flowers, it is of no use. Over here it is very, very useful. So usually these plants are brightly colored and they will have large showy flowers, and in species where the flower size is very small, all of these flowers will cluster together to form an inflorescence. So they will appear nice to the insect and attractive, so that the insects are attracted to the flower. Another way flowers can attract insects is by producing some sort of odor. If it produces like pleasant odors, then bees and butterflies will be attracted to it. If it produces unpleasant, foul odors, then flies will be attracted towards it. Either ways, it will get the pollinator come to the flower. But in order to ensure that they keep visiting and bring about pollination, rewards have to be given. Rewards are given in the form of nectar and pollen grains. So when they come to the flower, they are given food in the form of pollen grains. They are given juice or some sort of drink in the form of nectar, so that they keep visiting the flower. And also in some flowers, you can see that in the flower there will be space provided for the insect to lay eggs. It will come to lay eggs, and in turn it will end up pollinating the plant. So that is there. So here the flowers are large and colorful and fragrant and rich in nectar. Even if the flowers are small, they are clustered in an inflorescence. Animals can be attracted to flowers either by color or by its fragrance. So if it is pollinated by flies and dung beetles or beetles, flowers will secrete odor, foul odor, so that those animals are attracted. Okay. Yeah. The rewards that I told you, it will give nectar and pollen grains. Sometimes it will allow the the insect to lay eggs inside. Okay, it will provide a safe place to lay eggs. Example, Amorphophallus, it is an angiosperm plant. It will produce the largest flower in angiosperms. This is the flower. Inside this, insects are allowed to lay eggs and they bring about pollination. And you also have a species of plant called yucca and a specific species of moth called yucca moth, which will bring about pollination in this plant. And this flower provides a safe place for this moth to lay its eggs. Okay. And sometimes some insects come, they take the pollen, they drink the nectar and they leave without bringing about pollination. They are called nectar or pollen robbers. Okay. Yeah. So that's about the different agents used for pollination. Now let's talk about a concept called inbreeding depression. Similar to how if continuously, marriages are happening within the same family, the offspring will be affected. They could be mentally retarded, they could be deaf, they could be dumb. So such things can happen in humans. You may have seen this in some of your close circles, relatives or friends. Whenever two very close relatives get married, then the chances of them having a child which has genetic disorders is much higher. Something similar happens in plants as well. If continued self-pollination keeps happening, then a process or a phenomenon called inbreeding depression could happen. What basically happens in inbreeding depression is the offspring that is formed, it is not as good as either parents. It will not be very productive, and it will not, it may not be very, viability also is reduced. Then we call it inbreeding depression. So plants have realized that similar to us humans, whenever they make babies, whenever new seeds are formed and when a new plant is forming, their baby plant is forming, they want the best for their children, right? Everybody wants the best for their children. So plants also have realized, if I keep on undergoing self-pollination, then the babies that I'm not going to make are not going to be very good. If they are not very good, then their children are not going to be very good. And ultimately, our species only will suffer. So they have, over the course of time, during the course of evolution, come up with so many devices that ensure or kind of encourage cross-pollination over self-pollination. Such devices that plants have evolved in order to try and reduce self-pollination and encourage more cross-pollination are called outbreeding devices. There's 4 outbreeding devices that's mentioned in your textbook. The first one is there is no synchrony between pollen release and stigma becoming receptive. When the pollen is released, the stigma is not receptive, or when the stigma is receptive, the pollen of the same flower is not yet released. When there is no synchrony, then even though the flower is bisexual, self-pollination cannot take place, because when the pollen is mature, stigma is not receptive. If the stigma matures first, at that time the pollen grains are not yet released. Okay, so that is the first point. The second point is the anther and the stigma are placed at such positions in the plant that self-pollination cannot happen. The chances of self-pollination is highly reduced, and mostly cross-pollination will take place. The third thing is, something called self-incompatibility. It is basically a genetic mechanism. If a pollen grain from the same plant falls on the stigma belonging to the same plant, the stigma will not even allow that pollen to germinate. It will not allow the formation of pollen tube and reject it right there. So that is another way of, ensuring that cross-pollination takes place. The last type is making the flower itself unisexual. Okay, so if the flower has only stamen or has only gynoecium or pistil, there is no chance of autogamy, right? Of course, geitonogamy can happen if they are unisexual and monoecious, if there is a separate male flower and a separate female flower present on the same plant, autogamy cannot happen, but geitonogamy can still happen, as in the case of banana and maize and all of those. But in case of papaya, you can see how there is a separate male plant, separate female plant. In such cases, when the plants are dioecious and they produce unisexual flowers, autogamy also cannot happen, geitonogamy also cannot happen, only xenogamy can happen. Alright, so you can see that here. Yeah. If it is monoecious, as in the case of castor and maize, autogamy can be prevented, geitonogamy cannot. If it is dioecious, as in the case of papaya, both autogamy and geitonogamy can be prevented. Now, let's talk about pollen-pistil interaction. What is pollen-pistil interaction? I told you how after the pollen grain has been released, it has to go and land on the stigma and it will form a pollen tube. Now, when the pollen goes and lands on the stigma, it is possible that on the same stigma, pollen grains from multiple, different species of plants would have landed, because if it's going by wind, it is possible for pollen grains from some other plant to go and land on the stigma. Or even if an insect is visiting, it probably visited the flower of one species and then it goes and visits the flower of another species, then it's possible to carry pollen from a different species. In such cases, pollen tube formation will not be allowed because it does not belong to the same species. And even if that plant shows self-incompatibility, if a pollen grain has fallen from the same plant, it will not allow that pollen to germinate. Okay. So before pollen germinates on the stigma, there has to be a dialogue that takes place between the pollen grain and the stigma. There are chemicals present on the surface of the stigma. There are chemicals present on the surface of the pollen grain also. An exchange of chemicals will happen. They communicate by means of chemicals, and it is the pistil's job ultimately to decide whether the pollen grain has to be accepted or rejected. After this dialogue between chemicals, the stigma will decide if the pollen is accepted. If it is accepted, it will allow the pollen grain to form the pollen tube. If for some reason, because of self-incompatibility or because the pollen belongs to some other species, the stigma can reject that pollen. If the stigma rejects the pollen, then the pollen grain will not be allowed to form the pollen tube into the style. Okay. So, if the pollen is suitable for that plant, it is called compatible pollen. If the pistil or stigma specifically comes across a compatible pollen, it will accept it and allow it to germinate and form the pollen tube. If the pollen that falls on it is not compatible because it's of a different species or because of self-incompatibility, then it will reject that pollen and not allow it to form the pollen tube. So how this happens? It happens because of a continuous dialogue that occurs between the pollen grain and the stigma by the chemical components that are present on the surface of both these. Okay. Now, what happens during pollen-pistil interaction? I'll tell you all the events that take place from the time the pollen grain falls on the stigma to the time the pollen tube enters the ovule. All of the events that take place together, we call it the pollen-pistil interaction. Okay. Now, pollen grain has fallen. Let's say it is compatible. These ones are compatible. These ones aren't. So these are not allowed to form a pollen tube. The others are allowed to form a pollen tube. Some of these pollen tube will start growing through the style, and eventually they will grow through the wall of the ovary and ultimately enter the ovule via the micropyle. The pollen tube that enters first into the ovule via the micropyle will be the one that will bring about fertilization. Alright. So what happens during pollen-pistil interaction is the pollen tube will form through the style, through the wall of the ovary, enter the ovule through the micropyle. Once it enters through the micropyle, the filiform apparatus that I mentioned earlier, which will be present in the synergids, right? It will first enter one of the synergids. When this pollen tube is forming, I'll tell you what happens. If this is the X sign of the pollen grain and this is the intine of the pollen grain. There will be some regions in the exine where sporopollenin will be absent. That is called germ pores. Through the germ pore, the pollen tube will form. Inside the three-celled pollen grain, you had a large vegetative cell that itself is contributing to the formation of pollen tube. Inside that, the two male gametes will be traveling. Okay, so as this gets longer and longer, the male gametes will start moving towards the tip. Okay, so once the pollen grain enters the ovule, the pollen tube enters the ovule through the micropyle, it will be guided by the synergid into the embryo sac. Once it enters the synergid through the filiform apparatus, it will break open and release its contents, which is the two male gametes. Of the two male gametes, I'll tell you how fertilization will happen. For now, just understand, it enters the embryo sac through the synergids, and in the synergids towards the micropylar end, you have this filiform apparatus. This is what will guide the pollen grain into the embryo sac. Alright, so once it enters the synergid will get disintegrated, degenerated, and the two male gametes will get released. Okay, so this is one male gamete, this is the second male gamete. Now all of these events, from the time the pollen deposited on the stigma until the pollen enters the ovule, all of this is called the pollen pistil interaction. Up until this point, we've learnt about the pre-fertilization structures and events. Now we have to talk about how fertilization happens. Because in the embryo sac right now, I have the male gamete also. So the male gametes are also here. And the female gamete egg cell is also right here. So fertilization has to be brought about. Fertilization in angiosperms is a characteristic feature. We call it double fertilization because two fusion events take place. What happens in the first fusion event is very similar to how fertilization happens in most living organisms. Over here, syngamy takes place. The haploid male gamete will fuse with the haploid female gamete to form a diploid zygote. This diploid zygote will eventually become the embryo. That is called syngamy. So what happens in syngamy? One male gamete, this one male gamete fuses with this egg cell to form the zygote, which is diploid. What happens to the second male gamete that is here? The second male gamete will fuse with the polar nuclei. Remember, in the embryo sac there were two polar nuclei. These two polar nuclei first fuse to form something called the secondary nucleus, and to that the second pollen grain will, the second male gamete will fuse to form a triploid structure called primary endosperm nucleus. So this process is called triple fusion. This is also a fusion event where the second male gamete, which is haploid, will fuse with the two polar nuclei to form a triploid structure, which is called the primary endosperm nucleus. Eventually that will divide and give rise to the endosperm. So during double fertilization, syngamy is happening, its product is the zygote, it develops into the embryo. Triple fusion is happening. Why are we calling this triple fusion? Because three haploid nuclei are involved in it. The one male gamete and two polar nuclei, all of them are haploid, and will give rise to a triploid primary endosperm nucleus, which will undergo mitotic cell divisions to give rise to the endosperm. So both of these fusion events will happen simultaneously. The formation of zygote, the formation of primary endosperm nucleus will happen simultaneously. But the primary endosperm nucleus develops into the endosperm faster compared to the development of zygote into the embryo. That is what is given in NCERT textbook. Endosperm development happens sooner than the embryo development. Why is that so? What is the function of the endosperm here? The endosperm is nutritive. Endosperm is utilized for the development of zygote into the embryo. This zygote can develop into the embryo only when nourishment is derived from the endosperm. So before the zygote can develop into the embryo, this nutritive tissue should have developed, right? That's obvious. Unless this is there, for this development, nourishment is not available. This makes nourishment available for the development of embryo from the zygote. Alright, so this has to form first and then later only this will form. when this forms, if this is available, it can derive the nourishment from that and undergo development, right? All right. Now let's talk about the endosperm. I've told you that endosperm forms first. Endosperm, as we know, is triploid in nature. It is nutritive, therefore it is filled with reserve food. It is used to nourish the developing embryo. I've told you about that. Initially, when development of endosperm happens, whatever primary endosperm nucleus is there, which is triploid, it will undergo free nuclear divisions. Nuclear division karyokinesis is happening, immediately cell wall formation will not happen. So within the same cell, you will have many nuclei present. This is called the free nuclear endosperm. Further during development, cell walls will form, cell walls will be laid between these nuclei and result in the formation of cellular endosperm. Okay. So in most common type, primary endosperm nucleus undergoes successive nuclear divisions and give rise to the free nuclear endosperm. Eventually, cell wall formation will occur and form the cellular endosperm. If you take coconut for example, tender coconut water, whatever is there, that is the free nuclear endosperm. After cell walls are laid in the free nuclear endosperm, whatever kernel forms, the white color thing inside coconut that we grate and use to make kobri mithai, that is called the what do you call it? Cellular endosperm. Okay. Now, endosperm may be consumed completely by the developing embryo, or it may, some amount may remain after maturity. If it is, examples are given. If it is completely consumed, example you see in pea, mostly in dicots, pea, groundnut, beans, it is completely consumed. Mostly in monocots, you can see that it will persist. Okay, also in coconut and castor, it will persist even in mature seeds. That's about the endosperm. Now let's talk about the embryo. Embryo is formed from the zygote. Zygote is a single cell that is diploid. This will start undergoing mitotic divisions and first form what we call the pro-embryo, and after that there is a stage where the embryo proper forms a spherical structure which is called the globular embryo. Here this is called the suspensor. This suspensor will not give rise to the future plant. This part of the embryo will give rise to the future plant. This suspensor makes sure that the embryo is penetrated into the endosperm so that nourishment can be derived from it. After the globular embryo stage, you have the heart-shaped embryo stage, and eventually you have the mature embryo. All right. Now, let's talk about the structure of a dicot embryo and a monocot embryo. A dicot embryo obviously will have two cotyledons. Monocot embryo will have one cotyledon. In all embryos, you will have an embryonal axis like this. Okay. To the embryonal axis at one point, the two cotyledons will be attached. Right now at this point, this is one cotyledon that is attached, and over here the second cotyledon is attached, right? The point or the part of the axis, I'll draw the axis in blue color. Okay. So the part of the axis that is present above this point of attachment, this part, that is called the epicotyle. The part of the axis that is present below the point of attachment, that is called the hypocotyle. The tip of the epicotyle is called the plumule. The tip of the hypocotyle is called the radicle. The plumule will eventually give rise to the shoot system, and the radicle will eventually give rise to the root system. Remember, this is the embryo, right? Embryo is going to give rise to the future plant. This is all happening inside the ovule wall. It is all going to happen inside the seed. This structure is present in the seed. When the seed germinates, it is this embryo that will give rise to the future plant. So the plumule portion of it will give rise to the shoot system, and the radical portion will give rise to the root system. All right. Now, let's learn about monocot embryo. Monocot embryo also has an embryonal axis. There are some differences here. This is the embryonal axis. In monocot embryo, you have only one cotyledon. This is the one cotyledon that is there. It is large and shield shaped, that is called the scutellum. The part of the axis above this point of attachment is called the epicotyle, below it is called the hypocotyle. The tip of this is called the plumule and the tip of this is called the radicle. Plumule is going to give rise to the shoot system. Now what is different here, additional here, is that covering the plumule are sheath-like structures that are present, which we call the coleoptile. Covering the radicle, you have a sheath-like structure that is called coleorhiza. You don't find coleoptile and coleorhiza in dicots, but you find them only in monocots. Over here you can see there is a small blub-like structure present here, which is labeled the epiblast. Epiblast is the highly reduced cotyledon. In dicots, you have two cotyledons, right? One cotyledon got highly reduced and in monocot, so they have only one proper functional cotyledon. This is the cotyledon that is highly reduced right now. It is vestigial, not functional. So this kind of indicates that monocots evolved from dicots. Okay, so that is your epiblast. Now let's learn about a process called artificial hybridization. If you are a farmer or a plant breeder and want to bring two characteristics present in two different plants in one plant. For example, let's say this is one variety, these are two varieties belonging to the same species. In this variety, very brightly colored flowers are produced. These are very commercially, it brings a lot of money to the plant breeder. And also each plant produces a lot of flowers. In contrast, this plant variety belonging to the same species produces flowers that are not so brightly colored, not much in demand, and it produces very few flowers. Alright, now this plant has some feature which is good for plants, which is it is resistant to pests, whereas this one is not resistant to pests. This is producing high yield, which is a good thing. That is not producing high yield, which is not a good thing, right? Now this plant variety has one good feature, high yield. This plant variety has one good feature, pest resistance. I want to try and bring both of these features in the same plant by producing a hybrid between these two plants. So what are the different steps that I will have to take? Okay. Now, first thing is I have to designate one plant as male and one plant as female. I will assume that this plant, I will make this female. I will think this plant is male. Okay, for my convenience. Let's assume both of these plants are bisexual. That means in both of them anther and androecium and gynoecium will be present. If I want this plant to be female and I'm saying that this is a bisexual flower, that means it has both stamen and stigma. If I want to make it female, then what should I do? I should remove its male part, right? So I'm going to cut off this male part and remove it. The process by which I'm going to cut and remove the male part of the plant, which is the androecium or the stamen or the anther or whatever you call it, that step is called emasculation. Now I want to make sure that the pollen grains produced by this flower only should go and land here. Okay. I do not want this pollen to fertilize this, so I cut and removed the anthers even before they became mature. I emasculated them even before they became mature. Now I want to make sure some random pollen should not come and fall here. So what will I do? Immediately after removing the anther and emasculating it, I will tie a bag around it. This step is called bagging. And once the stigma becomes receptive, I will specifically cut this anther, go and dust the pollen on top of it and tie a bag around it again. That is called rebagging. That ensures that whichever pollen I am interested in, that is only going and depositing on the stigma. Now, I will allow pollination to fertilization to happen and the seeds to form. Fruit will develop in the bag only. After that, I will take out the seeds and sow them, plant them and get the hybrid. In the hybrid seeds, in the hybrid plants that you get from the seeds of this artificial hybridization, some of the plants may probably show both of these characters together in the same hybrid. It may happen, it may not happen. It's also possible that this feature and this feature may come together in one hybrid, right? So that is also possible. So what are the different steps that we took here? Firstly, emasculation, and emasculation is done only if it is a bisexual flower. If it is a unisexual flower, there is no need obviously. If it's a unisexual flower, it will only have either the androecium or the gynoecium. So female flower is already designated, right? So here, removal of anther before it dehisces is called emasculation. It's not essential in unisexual plants. After that, we cover a bag to prevent some random pollen from falling on it and contaminating it. And then once the stigma becomes receptive, you will dust the pollen of your choice on the stigma and allow, tie a bag around it again, which is called rebagging. After that, fruits will develop within the bag. You will collect the seeds and sow them and look for if the plants have your choice of or your combination of characters present. All right, that is artificial hybridization. Now, fertilization is over. After fertilization, whatever structures formed, embryo formed, endosperm formed, from the zygote, embryo formed, from the primary endosperm nucleus, endosperm formed. After fertilization, ovule will transform into the seed and the ovary will mature to form the fruit. Okay, so these are all post-fertilization structures and events. Seed is the final product of sexual reproduction. An ovule after fertilization will become the seed. So seed can also be described as a fertilized ovule. Where does it form? Obviously inside the fruits. What are the different parts that you can see in a seed? In a seed, you will have outer protecting seed coats. The integuments present in the ovule itself will transform into the seed coats in the seed. There will be cotyledons present. If it's a dicot, there will be 2. If it's a monocot, there will be 1. And there will be an embryonal axis to which the cotyledons are attached. We've learnt about it in the embryo structure, right? Now, depending upon whether in mature seeds, endosperm is present or not, you have 2 types of seeds. If endosperm is present at maturity, we call it albuminous or endospermic seeds. If endosperm is absent at maturity, the embryo has fully consumed it, then we call it non-endospermic or exalbuminous seeds. Okay. So example for non-albuminous is pea and groundnut. Here endosperm is not present at maturity. Albuminous example you can see in monocot plants mostly. You can see it in castor also. So in mature seed you will find wheat, maize, barley which are monocots and also castor which is a dicot. There you can see even at maturity some amount of endosperm will be retained within the seed. Alright. Yeah. Generally what happens is whatever nucellus was present inside the ovule, after fertilization, when embryo development happens, endosperm development happens, the cells of the nucellus will disintegrate, okay? But in some species like in beet and also in black pepper, the nucellus remains as a thin layer in mature seeds as well. That is called the perisperm, okay? So whatever residual persistent nucellus is found even in the mature seeds, that is called the perisperm. All right. Now, in the seed, the integuments form protective seed coats. The micropyle will remain as it is. I showed you in a diagram before also. Because of the presence of micropyle, whatever gases are required for respiration, they can enter the seed. And also during germination, water is required. Water can enter the seed during germination through this micropyle. As the seed undergoes maturation, it will get dehydrated, it will use a lot of water and its water will come down to just around 10 to 15% of its mass. That's just the amount of moisture that mature seeds will have. As a result, overall metabolic rate of the seed, of the cells in the seed will go down. Sometimes when the conditions are not favorable for germination of the seed can become dormant. It will be inactive. It will not germinate, okay? So that is called seed dormancy. As and when the conditions become favorable for seed germination, dormancy will be broken and the seed will germinate, okay? There are many advantages that seeds offer to angiosperms. Many fruits even have special mechanisms for dispersal of seeds to new habitats. If any angiosperm has to spread in distribution. Right now it's found only in one area. If it has to get distributed and expand its range geographically, then they have mechanisms for it. There are some species of plants where the fruits will open by itself if it is not plucked or anything, like lady's finger and all. If you leave it as it is, they have mechanisms to open and disperse the seeds far off. And in fruits that are fleshy, for example, if a bird goes and feeds on a fruit in one tree and it has consumed some seeds, it goes elsewhere and it excretes there, it poops there. The seed comes out in its excreta and it can germinate there and it can germinate and form a new plant in that new locality. So they have mechanisms to ensure that the seeds are dispersed. In order to, give reward to animals that come to it and help in dispersing seeds, fruits are formed, right? Animals come to eat fruits and in turn help in the dispersal of seeds. Yeah. See, because in the seed you have sufficient food reserves in the form of endosperm and cotyledon, even up until the seed germinates, produces leaves and starts photosynthesizing, its nutritional needs are taken care of. They have two very hard seed coats that protects the embryo that lies within it. Because it is a product of sexual reproduction, newer genetic combinations arise. Seeds are the basis of agriculture, and one very important property of the seed is its dehydration and dormancy. If the seeds did not undergo dehydration, it would have been prone to a lot of microbial infestations. And also, if it did not become dormant, then we could not store seeds for a long time and use it repeatedly for agriculture. We store rice grains at home today. We store wheat at home today for several months. If a seed germinated as soon as it was formed, then it would not be possible to store them as food for long periods and use it for our, daily nutritional requirements, right? So because of the property of dehydration, infestations are less. Because it is dormant and does not germinate immediately, it can be stored for long periods of time at homes for food and also by farmers to use in the next cycle as seeds for getting new plants. Alright, so these are some advantages that seeds offer angiosperm. Similar to how we learnt about pollen grain and its viability, there is viability for seed also. The period during which a seed retains the ability to germinate, it's called seed viability. Some seeds are shown to be viable, have retained viability for as long as 10,000 years as well. It differs from one species to species. There are two examples given in NCERT. There is one plant called Lupinus articus. This was excavated from the tundra region where it's very, very cold and of course there's a lot of ice and snow. The seeds of this plant was buried under the snow for a really long time, for 10,000 years. After they found it and they thawed it and they germinated it, this seed was 10,000 years old, but it, after it was sown, it did germinate and produce flowers as well. Another example is Phoenix dactylifera, which is our date palm. A seed that was 2,000 years old was discovered during an excavation from near the King Herod's palace near Dead Sea. This also germinated after an estimated 2,000 years. So some species of seeds have the capacity to retain their viability period for such long. All right. Now let's talk about the fruit. This is also a post-fertilization structure that forms. After fertilization, as the ovule transforms into the seed, the ovary simultaneously will transform into the fruit. Okay. Depending upon whether the wall of the fruit is fleshy or dry, you have two different types of fruits. If it is fleshy, like in the case of let's say mango and orange, guava, it is called a fleshy fruit. If it is dry, as in the case of groundnut, mustard, it is called a dry fruit. Depending upon which part of the flower is involved in the formation of the edible part of the fruit, we have two different categories. We have true fruit and false fruit or pseudocarp. We call a fruit a true fruit only when it has developed from the ovary. In some cases, like strawberries and apple and cashew nut, you can see how the fruit develops from the thalamus. The edible part of the fruit develops from the thalamus and not the ovary. In such cases, we call it the false fruit. Okay, so true fruit develops from mature ovary, false fruit develops from other parts, any part other than the ovary like the thalamus. Examples, remember, apple, strawberry and cashew. Alright, now, yeah, here you can see this is an NCERT illustration. What you see here, this is the actual ovary. What we eat, the fleshy part, is actually the thalamus. Okay. Yeah, now let's talk about parthenocarpic fruits. Parthenocarpic fruits are seedless fruits. When a fruit forms without undergoing fertilization, then the seeds will not form in it. The fruits will be seedless. This can happen naturally, as in the case of banana, or it may be induced with the help of plant growth regulators as well. So parthenocarpic fruits are fruits formed without fertilization. They will not have any seeds as a result. Okay, talking about apomixis. Apomixis is a phenomenon that we see in certain angiosperm families like Asteraceae and grasses. Apomixis is a type of asexual reproduction. You know that in asexual reproduction, formation of gametes, fusion of gametes will not take place. Here, we learned that seed is a formation of seed is a product of sexual reproduction. Apomixis is a type of asexual reproduction that tries to act like sexual reproduction. It kind of mimics sexual reproduction. In sexual reproduction, seed formation is there. In asexual reproduction, seed formation is not there generally, right? But in apomixis, seed formation happens even though there is no formation and fusion of gametes. Therefore, it is a type of asexual reproduction that is mimicking sexual reproduction by producing seeds. Okay, so yeah. Here, seeds are produced without fertilization. If fertilization itself does not happen, then how can you say that it is a product of sexual reproduction? You can't. So, apomixis is asexual reproduction which mimics sexual reproduction. Many different ways are there by which apomictic seeds can be obtained or developed. One example is, in some species, egg formation. During egg formation, meiosis will not take place. The egg is formed, the egg that is formed will be diploid. This diploid egg itself will develop into the embryo. So, meiosis is not involved, fertilization does not take place. A diploid egg itself will act like the embryo, and embryo is formed within the seed. That is called apomixis. This is very beneficial to farmers because some farmers will have to buy hybrid seeds every time from the market, because when you allow a hybrid plant to undergo sexual reproduction, there will be changes in the characters in the seed, because you know newer combinations will arise every time sexual reproduction happens during meiosis. So even though you have your desired combination of characters in one hybrid plant right now, if you allow that to undergo sexual reproduction and obtain the seeds from that plant, there are chances that the seeds of that plant will not have the same combination of characters, right? So if you want the same combination of characters again, you'll have to go to the market and buy hybrid seeds year after year, season after season. But if there is a way we can figure out how to make hybrid plants produce apomictic seeds, seeds are formed, there is no sexual reproduction, seeds will retain whatever characters are there in the parent. The farmers will not have to spend money to buy seeds season after season. That will save them some money and cost. So that is one thing that is under research currently. Last concept of the chapter, polyembryony. Poly means many. Embryo obviously refers to, embryony obviously refers to embryo. Generally in plants, within one seed only one embryo will be present. But in some cases, like in mangoes and citrus plants, one seed can have more than one ovary, more than one embryo. If there is more than one embryo present in a seed, we call it polyembryony. It can be found in citrus and mango. What happens here? How does the other embryo form? Of course, one embryo has formed because of sexual reproduction from the zygote. How do the other embryos arise? Some of the cells in the nucellus itself, which are diploid, can start behaving like embryos and form embryos. The nucellar cells surrounding the embryo sac will start dividing. It will protrude into the embryo sac and develop into the embryos. And in such cases, within the same seed, you will have many embryos. Sometimes the cells of the integument also can develop into the embryos. So that can happen. So within the same seed, when you have multiple embryos, from the same seed, you can get many saplings. Alright, this is generally seen in citrus plant seeds like oranges and lemons, and also in mango. With that, we have come to the end of this chapter. I hope this session was useful. If it was useful, kindly let me know in the comments. If you have any doubts, please feel free to ask them. I will answer them as soon as I can. I will see you in the next class with the next chapter, Principles of Inheritance and Variation. Time is ticking. Exams are coming closer and closer. Do not procrastinate and study every day and study hard. All the very best. Thank you.